Okay, well, hello. My name's Dr. Andrew Morokoff. I'm a neurosurgeon here at Royal Melbourne Hospital and the University of Melbourne, and I'm involved in our research project on brain tumours. So I run a lab here that has a number of different interests in looking at brain tumours, which is one of the worst types of cancer that people can get. It kills a disproportionate number of young people, and many people with high-grade tumours unfortunately only survive a year or less. So we're really committed to finding new ways of treating tumours and new ways of managing patients with brain tumours. So a few of the research projects that we work on are to do with understanding the molecular signalling within brain tumour cells, understanding how brain tumours cause epileptic seizures, for instance. But one of the projects I've been most excited about recently is trying to find what's called a biomarker for brain tumours. So a biomarker is anything that we can easily test in the body that can give us information about diagnosing the tumour or predicting how it's going to behave in different people, or possibly helping us to decide what sort of treatments are best for it, so-called personalised medicine. So for brain tumours at the moment, a patient comes in with a suspected brain tumour. The way that we diagnose it is by doing an MRI scan. So an MRI scan is a very complex scan. It takes about 45 minutes. It's expensive. It's inconvenient for the patient. It can be quite uncomfortable, especially if you're claustrophobic, because it requires lying in a small tunnel. But it gives us very good imaging information about the tumour, and it helps us diagnose with fairly good accuracy what type of tumour it's likely to be. But the ultimate diagnosis is achieved when we do surgery on the tumour. We do neurosurgery, so we do a craniotomy, which involves making a hole in the skull. It's usually an operation under general anaesthetic that takes 3 or 4 hours or more. And then we take part of the tumour and we send it to the pathology lab, and they examine it under the microscope and tell us exactly what grade of tumour it is or what type of tumour it is. So that combination of MRI and surgical operation with biopsy is really the only thing we have at the moment to tell us what we're dealing with in terms of the patient's tumour. And then after the patient's had surgery, they often go on to chemotherapy or radiotherapy treatment. And the only way we have at the moment to know whether their tumour is responding to that or not is to do another MRI. So most patients come in about every 3 months to have repeated MRI scans to follow the tumour along and see if it's shrinking or if it's progressing. So it'd be really fantastic to have a new way of following along what's happening to the tumour without having to do an expensive and inconvenient MRI. So our image, our dream is that a simple blood test could even be done in, for instance, the local GP surgery, particularly for country patients or interstate patients, and the sample could be sent to us to analyse, and we could give much more accurate information about the likelihood that the tumour is responding to the treatment or the potential that it might come back. So the last couple of years we've been working very hard on trying to identify what we can look at in the blood test to tell us about the status of the tumour. So there are a number of different possibilities. We started off looking at circulating cells in the blood. So it's quite amazing that everything that happens in our bodies really ends up in the bloodstream. Cells break off and float around in the bloodstream, or bits of genetic information like DNA or RNA circle in the bloodstream. And we can test for those. So we've looked at circulating cells, particularly cells that come from the endothelium, which is the lining of the blood vessels. We've also looked at DNA to try and detect specific DNA mutations that come from brain tumours. There are a number of known DNA mutations that we know about that occur in brain tumours. But the problem with that is that they're only in about 20 or 30% of brain tumours, because there are, even in the same type of brain tumour, there's a lot of heterogeneity or variation in the various gene mutations. So if we find one gene mutation, we can say for sure that it's from a brain tumour, but we're only going to find that in about 20% of patients, so it's not a very sensitive test. So recently we've been looking at what are called MicroRNAs, which are small pieces of RNA that are in every cell. And so far we know that there's about 2,000 of them that are present. So they are produced by our normal DNA genome, but they're not RNAs that are used to go on to make protein in the cell, they actually regulate the other RNAs and the other DNA system to have different regulatory effects on the cell. So they might upregulate the growth of the cell, or they might downregulate it, or they might promote the cell to go towards an early death. So there's a lot of different functions of microRNA. And our idea is that the microRNA can be detected in the blood by the blood test, and by looking at the profile of what different microRNAs are up or down, we can get information about the brain tumour. So we've done this now on 91 patients to do a preliminary test to see if this can work. And we have blood samples from before their operation and then after their operation, up to about 10 different blood samples, depending on how long they are treated for. And at the moment, we've looked at the preoperative blood samples and found which particular microRNAs are up or down regulated in high-grade and low-grade tumours. So it's very clear that if we take tumour patients, and we've also taken blood from healthy patients, that the microRNA pattern is extremely different between the patients with a brain tumour and the patients without a brain tumour. So the tumour is definitely significantly affecting the pattern of microRNAs in the bloodstream, which is very exciting potentially for us being able to use this blood test as a way of following the brain tumours. There's also significant differences between what high-grade brain tumour puts out into the blood and what a low-grade brain tumour puts out into the blood. And what we're doing currently at the moment is looking at all of the post-op blood samples to see what differences there are as the patient responds or doesn't respond to the post-surgical treatment, for instance, the radiation or the chemotherapy. So how do we actually detect the microRNAs? Well, we have a fantastic new next-generation sequencing platform called the Nanostring Machine, which is able to very rapidly and very accurately detect 800 microRNAs out of about 2,000. So they're the 800 that are known to be involved with most cancers. So we can rapidly screen 800 microRNAs, and then we get we use bioinformatics analyses and statistical analyses. We use modern big data methods to look at these 800 microRNAs across 90 patients, and we're able to sort them into different patterns and to get information about what's happening in the patient's brain. So I think that's a very exciting new way of looking at the way we manage brain tumour patients, and we should pretty soon have a very accurate biomarker to be able to help us do this. Once we've narrowed down exactly which microRNAs are important and what the biomarker will consist of, we then need to go ahead and validate it in a clinical trial, a prospective clinical trial, which will probably run over the next couple of years and involve a few hundred patients. We also want to be able to make the testing of the blood very easy. So we're collaborating with our colleagues in the medical bioengineering department, the Centre for Neural Engineering, to put the microRNA test on a small chip. So our goal is to have it to be as easy as, for instance, a diabetic patient testing their blood sugar by just doing a quick finger prick test and being able to put it on a little chip, and they'll get an instant readout, perhaps on their smartphone or on their laptop, that can then be sent back through a secure data platform back to the clinicians, and that way we can follow the patients much more accurately, much more closely. Because it's easy to have a blood test, for instance, once a week. It's much easier and more accurate to do that than to have to come down to Melbourne to the hospital to have an MRI every 3 or 4 months. So I think this will be a great advance for brain tumour patients, and if it works, it would also be a great way of monitoring patients for every future clinical trial. So if there's a new chemotherapy drug that's being trialled or a new way of treating them, having this blood test would be a fantastic way of telling if it's going to work or not, and it could have application really throughout every cancer clinical trial in the world.